Hello again. This is Mauricio Bonifacino, and I'm going to introduce you in this presentation to the microcharacters in Compositae. So I will start with trying to propose a sort of definition for what microcharacters are, then provide some historical context on the use of microcharacters, a few quick tips on how to prepare the material in order to be able to observe those microcharacters, at least some of them. And then I'm going to present some examples of microcharacters. There's not a real definition of microcharacters. When people first starting to use, or at least in our time, these sort of characters, they were not very specific about the scope of the microcharacters, but I guess that it's pretty safe to say that microcharacters are those that can be seen under the compound microscope or higher magnification. There's no specification how powerful that magnification needs to be, but these are not characters that you're going to see with a naked eye or with a simple stereoscope. You definitely need higher magnification in order to see them. And here you have an example of the shape of the filament color in the stamens of the Cynicione, with the case of the baluster form filament. So in reality, the actual origin of the microcharacters, we can say that date back to Henry Cassini. He is the one who actually started, the practice of looking at minute details in order to find information to make statements about the systematics of the composite. He basically looked at microcharacters that he took from the styles and the anthers to define the different groups of the composite. However, in our modern time, the one who could say that caused a revolution was Harold Robinson, who sadly passed away last year. No, Robinson came to the composite from the world of bryophytes and is the absolute champion of the microcharacters. By means of using microcharacters, he, together with Robert King, splitted the mega genus Eupatorium that had more than 1,000 species into a myriad of smaller genera, with the end result that Eupatorium now it's just a few dozen of species. It's an interesting story how this bryologist got interested in the composite. He was introduced to them by Robert King, who took a plant that he just had collected in Mexico, I believe, and said to Harold, well, you're used to look at small things, look at this small composite and see if you could get something interesting. And that was a whole new world for Harold. And that led, I believe it was the next year or, a couple of years right after, to a revision of the genus Hofmeisteria. And in that revision is where they make some comments about the use of microcharacters, something that had been neglected till that moment. So I quote from that paper on the Hofmeisteria. He said, since some of the characters involved in this study have not been previously used in delimiting composite genera, and since some of the traditional characters is misleading, the following comments are offered. And they provide on that paper a thorough description of those traits, and they even illustrate some of them. Basically, they rely on corolla pubescence and the shape of endothelial cells in the anthers, the shape of the anther appendages, the cypsella pubescens, and the shape of the carpopodium. So using all that information, they were able to figure out some of the problems associated with that genus, and that will start a career that will extend over 10 years, in which they will split, as I said before, the megagenus Eupatorium, and that will give Harold Robinson and Robert King, the title of perhaps the biggest splitter that the plant taxonomy world has ever known. Just to give you a little bit of background, so here is an infographic that shows the published record of Harold, and there you can see where the Hofmasteria paper is. So the horizontal lines mark tens of papers. So you can see that he was very, very prolific through his career. In red here we have all the papers devoted to the Eupatori, and you see how the Hofmasteria paper is the start of a whole series of paper. Then in 1970 is when they published their paper, The New Synantherology, in which they present the microcharacters to the broader audience in a paper in Taxon. In that paper, they don't say very nice things about the traditional characters, and they praise, highly these microcharacters. A lot of people criticize them after that publication, but afterwards, Harold reminiscence of those critiques that they receive, and they say, well, the one thing they didn't criticize is that what we did was not new, because that what we describe as new synantherology was what was done since the very beginning by Henry Cassini. So at any rate, I just wanted to show this graphic because it showed the magnitude of the work done by Harold Robinson and Robert King with the use of microcharacters, which are the subject of this short introduction. So, what is the quick and dirty way to look at these microcharacters? Well, first, you either have fresh material or you need to, soak in water. Or basically you have to boil your dry herbarium specimens, the flowers that is, in order to see them. So you will boil them for about 5 minutes until you see that if you put a head in the beaker, that the head start to sink in the beaker. Then you will put your samples into diluted sodium hypochlorite. That is a clearing agent. Then you're going to wash it in water, and later on you could add a dye to enhance the contrast. You could actually, after you boil the sample, you could actually take the sample into the microscope and look at directly. But if you do this, you might get a better results, as you will have a clearer tissue and a more contrast. But keep in mind, though, that preparing the material this way will make the slide permanent. But this is a relatively quick way to prepare the specimens in order to see several of the microcharacters that I'm going to mention briefly in just a minute. Another way, it's Harold Robinson's way, which is to use the Hoyer solution following Anderson's formula. So Anderson was a bryologist, was the PhD advisor of Harold Robinson, and he had just published an article about how to better prepare the Hoyer solution in order to make slides to study bryophytes. Remember that even if Harold was, famous or is famous for the study of the composite, he started his career as a bryologist. So according to Anderson's formula, in order to prepare the Hoyer solution, you need 50 milliliters of distilled water, 30 grams of gum arabic in flakes. Anderson specifically advised against using the powder form. Then 200 grams of chloral hydrate, this is the clearing agent, and 20 grams of glycerin. So what are the benefits of the Hoyer solution? Well, it has more clarity and contrast because of the properties of the solution, and it's semi-permanent. And once it's dried, it gets like a hardened substance, almost like a resin. So that is a way in which you could prepare specimens and store them for, for the midterm in order to be able to go and return to them for further study. So with that out of the way, let's see which are one of the main micro characters. Definitely the microcharacter that has been most studied in the compositing, even if it has not been referred to as a microcharacter, are the trichomes, especially the trichomes in the reproductive parts. Here, in an illustration from Ramaya from 1962, we have different type of glandular trichomes, but we have all sort of trichomes varying in degree of if they secrete substances, the number of constituent cells, the shape, the size. There's a whole galore of terminology referring to trichomes and floral parts. Another interesting microcharacters is the ornamentation of the receptacle. So, here we have some illustrations by Roque and Funk from 2013, in which they show some type of receptacles ornamentations, and you can see on the left that in certain cases we have very conspicuous trichomes surrounding the places where each flowers were attached to the receptacle, and then you have different degrees of honeycombed structures on the surface of the receptacle. Depending on how tall the walls of that honeycombed structure are, we will have different terms. But this is just a sample of the different forms that could occur in that particular character. A sort of character that requires, higher magnification. These are images obtained through the scanning electron microscope, but in certain cases you can even see them through the compound microscope, are the ornamentations of the corolla surface. This is a work by Buggy from 1977, where the surface of the corolla cells, specifically the ones on the abaxial sides, are illustrated here. And this researcher, provided some interesting patterns across the family. So there you have an interesting source of microcharacters. Another sort of character that we could, label into the micro characters group will be the insertion of the filament in the corolla. How that insertion is. And this is from Robinson in 2009. From the Funk et al. Volume. So we have the filaments inserted above the distal expansion of the corolla. There you have on the left a tubulose corolla, and look how the filaments are inserted once the tube has started to broaden. Then we could have the filaments inserted at the point where we have an abrupt expansion of the corolla. In this case, they could be inserted at the base of the corolla. They could be inserted gradually along the tube, or they could be inserted at the base of the lobes. Those are different sort of situations that could occur. Well, then there's what I use in the introduction, the shape of the filaments and the thickness of the walls. You have the baluster form shape of the Anther color, typical of some Sinisioni. And then you have what is called the cylindric shape. The filaments in the compositae are not necessarily cylindric, are more like a flattened structure. But with this, what is meant is that it doesn't have a special thickening. Other character that you might want to check is the thickness of the cell walls that form the anther collar. That could vary, and that could also offer some interesting information. Also pertaining to the anthers, we have the anther apical appendages, the form, the size, whether if it's present or not, the color, all those are important characters, as we see here in this illustration from a paper from Grossi et al. From 2020, where they revisit all the microcharacters used in the Eupaterae. A very interesting paper that you should look for in search for all sort of microcharacters available in the Eupaterae. Very interesting paper. Then there's another interesting character derived from the androecium, that is on the cells of the endothecium, and it's particularly pertaining to the thickenings on the cell walls. So at least 3 patterns have been described. What is called radial, in which the cell wall thickenings are located all around the perimeter of the cell, as we see here on the image on the right. And the other type is the polarized type, in which the cell wall thickenings are on opposite extremes of the cell. There's a third type of endothecium cell wall thickenings, which is the transitional type, in which we don't see any special thickenings around the cell wall. Then we have the style ornamentation. That is the form of the collecting trichomes. That we could consider a sort of microcharacters, very useful, and in fact one that was used by Cassini himself. In this case, we have Acanthostylis bunifolius, a genus described by Harold Ramis and Robert King. And in the name of the genus, it's reflected the microcharacter that they use. Acanthostylis means spiny style. And if you look at the style, you will see a long and spiny collecting trichomes. Very, very unusual for the Eupaterae, and that what prompted these researchers to name the genus after that particular trait. Here we have a very nice illustration from Cabrera, from a paper by Cabrera from 1959, where he described the styles of the Barnardicea. So you can see there, without being referred as microcharacters, we see them there. Very detailed illustrations and descriptions of the shapes and the different ornamentation of both the style branches or the shaft. Another sort of interesting trait that has been labeled as a microcharacter is the pattern of phytomelanin in the fruits. The phytomelanin is a substance that is very dark, very blackish, and is deposited in the walls of the cypsellas in certain groups. So in this paper from Freitas et al. From 2015, they discover a hitherto unknown presence of phytomelanin in the Vernon eye. So that you see there of color black, that the arrows are pointing, that's the phytomelanin. And you see that phytomelanin in this particular case is attached to a layer of sclerenchyma that surrounds the cypsella. And that sort of distribution was unreported till the date of the paper for the compositae. So these authors propose this classification, this expanded classification of how the phytomelanin is distributed. So the way the scheme, I color the scheme to make easier to spot the differences, is with the yellow you have the epicarp, that's the outer layer of cells in the fruit. Then with orange you have the sclerenchyma, and with pink you have the parenchyma. So the type on the left is what is known in the eupatory. So you have that notice how the phytomelanin is always associated to the sclerenchyma, but in the case of the eupatory you have that between the sclerenchyma and the phytomelanin and the epicarp you have a layer of parenchyma. This is also the case in members of the Helianthi alliance, with the difference that the shape of the sclerenchyma is somewhat different than the one that presents in the eupatory. But notice the difference in the Vernonae. Look how cool this is. The sclerenchyma is right next to the epicarp. There's no intermediate layer of parenchyma, and that is the type that Freitas et al. Described in 2015. So very, very interesting result. The sclerenchyma on the Cypsela could also offer, the sclerenchyma itself could offer also some source of microcharacters, like in this paper by Sancho et al. From 2006, where the sheer size of the sclerenchyma layer was useful to differentiate the two species in the genus Microgene in the Asteria. In a paper by TICA et al. From 2020, where they studied the structure of the Cypsela wall, how the sclerenchyma and the parenchyma were associated. Well, look how interesting all the patterns that they describe for a grouping de Lactucci. Very, very interesting. Another source of microcharacters are crystals, and here we see tiny samples of the different sort of calcium oxalate crystals that Bertel Nordenstam described for the Cinicioni from the ovary wall. Very, very wide diversity of shapes and sizes of these crystals. And finally, here we have the Carpopodium. That sort of the foot of the Cypsela. These are the original illustrations from the King and Robinson Hofmeisteria paper. And so you can see that there's not only variation in the shape of the structure and how distinctive it is, but also in the shape of the constitutive cells in terms of in which way they are elongated or on how thick their walls are. Here is an image of a Carpopodium from the scanning electron microscope, where we can see very nicely the structure of the Carpopodium and its constitutive cells. We also see the twin tricone cell characteristics of the Cypsela of the Compositae, in this case in a genus named after Haroldia. But you don't necessarily need to go to the scanning electron microscope to see Carpopodium. You can see it even with the compound microscope, or sometimes even with the stereoscope, like in this case of a porophyllum in the Tagiri. And with this image of the very peculiar club-shaped awns with glandular trichomes for a pappus in the genus Adenostema, I thank you for your attention, and I hope you find the search for microcharacters as interesting and exciting as I do.